Hello, my name is Manny Pena, and today I will be discussing the determination of gender and the genes and hormones that influence gender. So, I am doing this presentation for the medical physiology course through University of New England online. Before I get into the determination of gender, it's important to talk about chromosomes and genes because they have a big influence on gender. So, chromosomes and genes are the recipe for making a living thing. And chromosomes are made up of DNA that's very, very tightly packed. So chromosomes are found in the nucleus of a cell. So as you can see in the diagram to the right, there is a cell with a nucleus where chromosomes are stored. And if we go deeper, we can see that these specific chromosomes are made up of genes. So genes are basically the ingredients of human life. And they basically can be known as special segments. And they're special because these segments code for certain proteins that give us our unique characteristics, such as different eye colors, different hair colors, different heights. There are tons of characteristics we could go through, but there are way too many. But it is important to note that these genes are going to be talked about later and how it influences the gender of a human. So there are 46 chromosomes in a human being, and there are 23 pairs. 22 of these pairs are autosomes, and one pair is a genosome, and it's also known as a sex chromosome. They're denoted as X and Y. The female chromosome has two X chromosomes, and the male has only one X and then one Y chromosome. So if you look at the picture to the right, the chromosomes that are highlighted in the red box are going to be the chromosomes that we are specifically talking about here. So, in the female, there is one inactive X chromosome, and it is inactivated in the form of heterochromatin. Heterochromatin is transcriptionally inactive, and it is the basis of genetic gender. And this occurs very early on in the blastocyst stage. And it's important to note, too, that the presence or absence of Y chromosome is going to be one of the determining factors of gender. And the reason for this is because the Y chromosome has a special localized gene that we'll get into. And this gene is known as the SRY gene. So, the SRY gene, as I stated in the last slide, is localized in the Y chromosome. And as you can see in the picture to the right, there is a Y chromosome and the SRY gene located at the top of this chromosome. So, the SRY gene works with other genes to form the testicle. So, it works in conjunction with autosomes and the X chromosome. So this means that this gene is present very, very early on in the early stages of embryonic development. So it works together to form the testes. And before I get into the testes and how they influence the determination of gender, it's important to note that there are two equivalent instructions present at the early stages of embryonic development. These two equivalent structures are the mesophrenic duct, also known as the Wolffian duct, that's present in males, and that is the duct that turns into specific male organs, such as the scrotum and testes. And then there is a paramesophrenic duct that's present in females, and it's also known as the Mullerian duct, and that's going to give rise to specific female organs. So at the early stages, we have these two ducts present. They're equivalent and they're still there. So, what causes one of them to stay and what causes one of them to go? So, the answer here is hormones. Specific hormones cause a regression of a specific duct, and that would be the paramesophrenic duct. So, going deeper now, development and degeneration of the particular ducts depends on the absence of two specific hormones. The hormones are testosterone and anti-Mullerian hormone, which is also known as AMH. So to go deeper in understanding what these hormones do, we could talk about the stages in male development. So, first step here is that the SRY protein binds to DNA, and then this turns gonads into testes, and then the fetal testes secrete hormones such as AMH, testosterone, and dihydrotestosterone. So, AMH is the hormone that is responsible for regressing the paramesophrenic duct. So as we can see in the picture to the right, we see that AMH is secreted by Sertoli, excuse me, there we go. It's secreted by Sertoli cells, and this then causes the generation of Mullerian ducts. So what this means is that this duct completely disappears, and female organs are no longer possible to be formed in this organism. Now, testosterone, on the other hand, is formed by Leydig cells, and this in turn converts the mesophrenic duct into specific components of the male. And as you can see in the diagram to the right here, it's probably blocked by the camera, but it is known as the Wolffian duct as well, and it forms vital structures for the male, such as the epididymis, vas deferens, seminal vesicle, and also the testes that go down to the scrotum from the abdomen. So it's important to note that in female development, if there is no testosterone or AMH, there is no mesophrenic duct. So in other words, there would be no duct that would allow for the development of male organs, such as the testes, which we just mentioned. So if this AMH testosterone, this AMH hormone is not present, then it would not regress the paramesophrenic duct, which contains the vital organs for females. And what does the paramesophrenic duct give rise to? It gives rise to the fallopian tubes, the uterus, cervix, and upper 2/3 of the vagina. So if there is no hormone inhibiting the development of this duct, then we will have female organs being developed. So unfortunately, there's a lack of information of genetic controls in female development. As far as we know, this is the primary mechanism that determines the gender in humans. And this concludes the presentation. Thank you very much for watching.